It's really a great pleasure, actually a privilege, really, to introduce our speaker today, Jake Brunkard. So it's an important talk for him. It's his TED talk. He has been in the department since 2020. So Jake has obtained his BA in biology and history in 2008 from Swarthmore College, and then a PhD in plant biology in 2015 from Berkeley. And there he worked on signal transport through plasmodesmata in plants in Pat Zambriski's lab. Then he moved for a short postdoc to Sarah Heck's lab at the USDA Plant Gene Expression Center. He was there as a postdoc for 2 years, and then he was able to secure funding, an NIH Early Independence Award, which basically allowed him to start his own independent research group over there. And he started a very productive research program on TOR signaling in plants, and more specifically, the role it plays in nutrient sensing in plants, and basically the downstream effects on plant development, growth, metabolism, and responses to environmental stimuli. He joined, as I mentioned earlier, he joined our department in 2020. So he was one of those lucky people who had to start up a lab while actually facing a COVID pandemic. And you can imagine that it's not easy. But he did amazingly well at that. And so in only a few years, really, he was able to develop a very productive research program to grow his lab quite substantially. So as an indication of how efficient he has been able to publish, since joining the department, 7 primary research papers, and that's in top-tier journals like Genetics, Plant Cell, the Plant Journal, Cell Reports, and so forth. 3 reviews, 1 patent application, and 5 additional manuscripts are in the pipeline. And these are all data, there is more at this point. Importantly, he was able to also secure substantial funding for his research. So he was able to get an NIH R01 grant, and that's really an amazing feat for a plant biologist. It's not very frequent. A supplement on that grant, a USDA NIFA as well, and 1 collaborative award from Wisconsin Partnership Program, as well as a number of graduate student fellowships. He really has become a leader in his field, and you can really figure that out by looking at the number of talks that he has given at national and international conferences, the fact that he was elected to organize a plasmodesmata meeting in 2024, this year in St. Louis. He served on multiple peer review panels for NSF, USDA, Slovenia Research Agency, and serves diverse editorial roles for 4 different journals, so Frontiers in Plant Physiology, BMC Plant Biology, Plant Journal, and Current Opinions in Plant Biology. So these are all really good signs that he's viewed as an international leader in his field. This is a tenure talk, and so I'm going to talk a little bit about his accomplishments in teaching, outreach, and service as well. Don't worry, that will not be too long, but it has been very remarkable, actually. So in terms of teaching, he has contributed to curricular development. So he spearheaded a restructuring of a plant genetics course to incorporate concepts of plant development. In addition to that, he has taught a new module in Genetics 701, 702 on domestication, I believe. And he started a brand new course for our graduate students in the genetics program, teaching how to write research proposals. That's course that has been highly effective. So it allowed basically the students on our program to increase by a lot the rate of success in fellowship applications from federal agencies and extramural funding. All right. So one of the interesting features about his teaching is that he incorporates techniques of active learning. He does that very, very efficiently. And he brings in an amazing level of enthusiasm as well. And so all of the students in the classroom are really very much both challenged by that, but also interested by the material. In terms of mentoring, he was able to develop a good research team, a large research team, with 1 postdoc at this point, 5 PhD students, and 8 undergraduate students. Importantly, they have been able to secure substantial funding, fellowships, basically, and also are productive in terms of publications. The fact that he's committed to excellence is really demonstrated by the fact that he actually participated in 2 different courses. One is the UW, the discussion project course, and he attended also the HMI Gilliam Program Laboratory Mentorship course. Okay. In terms of service, he has been very active with the genetics graduate program. He has served on almost every committee that exists in the program. The admissions committee, which is a lot of work. He chaired the retreat committee. He sits on the steering committee. He also contributed to program-sponsored workshops, such as on lab communication, mentorship, and responsible conduct of research. At the university level, he is serving on faculty search committee for 2 faculties, as a matter of fact, at the botany department, and also graduate research scholars admissions committee. At the national level, we already discussed that. So overall, his accomplishments, both in research, teaching, outreach, and service, have been really remarkable. And it's really no surprise that in 2023, he was recognized by a Vinus Early Career Award. So today he's going to tell us about the beautiful work he's doing on TOR signaling in plants. Please join me to welcome Jay.
Thank you, Patrick, for that very nice introduction. I appreciate it. Patrick teaches classes with me and has been this incredible mentor throughout the entire time since I've been here. And a lot of my success is thanks to his support, and that I can go over and knock on his door anytime I want to and ramble about whatever I want to ramble about. Sometimes science and sometimes my mood that day. So, I'm going to tell you today about our work on the target of rapamycin signaling pathway, which I'm trying to restrain myself on the puns, and so I think it's only one today, but conductor. It's the conductor that orchestrates plant development and metabolism. Throughout my talk, there's going to be references to this film Annihilation. If you walk away with one thing, just go watch the movie Annihilation. It's about mutagenesis and genetics. You need to go see Annihilation. You'll see it's going to come up repeatedly, but this is an end title scene from the movie Annihilation. That's also me restraining myself. I had a lot more movies I wanted to make reference, but I'm just focused on Annihilation. So, you know. Before I talk about the science, I wanted to briefly make some acknowledgments so that I make sure that I don't run out of time at the end to make them. So first, I have to make a big thanks to my Berkeley group, who joined my lab when I wasn't even a PI yet. And so pictured here are my graduate students in Berkeley, Michael, Snigdha, Nanticha, and Will, and my postdoc, Regina. And Michael and Regina were the first members of my lab and really got everything up and running for me. And then Michael continued on and was generous enough to join me in the move to Madison. And so he's here and continues to be really instrumental to all of our progress. Another one of my first postdocs was Jasmine Abraham Juarez. And she just published, we just published a paper together in Plant Journal, her first author paper, and she recently became a PI. So she's my first postdoc to go off and become a PI at Cinvestav in Mexico. And I don't have any time to tell you about our maze development work today, but we have a whole other set of things going on with maze development. So anyone who wants to hear about leaves sometime, please let me know. I'm happy to talk about that. And it was on the cover. It was on my first cover image. And then here's a recent photo of the group here at Madison, who I'm sure most of you know, Rory Greenouch and Katie Klimpel and Ryan Martinez, and Katie Lund and Aaron Albrecht are the PhD students. Allie is our amazing undergrad who keeps everything running in the lab. Yeah, it's been so great to have the lab grow quickly since we came here to Madison. I should say, so on the maze development, Michael just published our first paper in genetics. So, I have a paper in genetics, tenure, please. I'm ready. I'm a geneticist now. We've accomplished it. Showing some very classical kind of genetics approaches, looking at development of leaf initiation in maize and making crosses. And there's Michael hugging his corn because he loves it that much. Okay, so today I'm going to be talking mostly about the target of rapamycin. And so I want to start off with an introduction to this protein TOR that I like to think of. It's not that we're really studying the TOR protein itself, but it's an organizing principle of the lab. So it's a doorway that lets us study a lot of different aspects of biology and keep us focused on something. So we can say we work on TOR even if we move off into different directions. And I'm just going to give a brief introduction here. So rapamycin, which is the drug that targets TOR, so it's the target of rapamycin, was identified and after a slurry of research came about from the penicillin discovery. So of course, everybody here is familiar with the story that penicillin was discovered as a antimicrobial that's generated by a mold that kills off bacteria. And a lot of other antibiotics were developed from that. And so then a lot of groups wanted to find in the other direction, are there any drugs that can be used to kill off that are antifungals? And so in that sort of big search, in the 70s, a group isolated this strain of Streptomyces hygroscopicus, which makes this cute little curly Q, weird shaped bacterium. And they isolated it on the Easter Islands, which the first peoples called Rapa Nui. And so that's the origin of the name rapamycin. And this compound is a really potent antifungal. Yeah, and as with most small molecule things, of course, it turns out now that there's a lot of bacteria that make this all over the world in soils everywhere. And so it's really prevalent, but it was first discovered on Rapa Nui. And then I fortunately don't have to say much about this because Joe Heitman was here a couple of days ago and gave a talk. I hope everybody got to hear that. And he and Michael Hall discovered that the target of rapamycin is this protein kinase called TOR, using a phylogenetic approach. So TOR is a protein kinase that's conserved across all eukaryotes, and it has a couple of accessory proteins called Raptor and LST8, and together they form a complex called TORC1. And TORC1 is found in effectively all eukaryotes, and in all eukaryotes, it's basically serving the same function. It's monitoring nutrient availability, especially the building blocks of life, amino acids, nucleotides, and by energy, I mean maybe carbohydrates, maybe ATP. There's a little bit of variation there, glycolytic intermediates. But just keeping track of those ingredients. And when all of those are available, the TOR complex is active and promotes growth and development. And again, it does that through many different means, but especially by promoting ribosome biogenesis. The TOR complex, these core components are so well conserved that they're almost identical in humans and plants. I always say that I would like to do an experiment of swapping them out. We are putting the human TOR into plants. I don't have any human volunteers to swap out their TOR with a Arabidopsis TOR, but if anyone ever wants to do that, I'm ready. I'm game. So of course, many of you are familiar with TOR, which is after Joe Heitman discovered it, then the groups that found it through more biochemical methods in mammals decided to call it mTOR, so it would be mammalian TOR, but it's the same. It's TOR. TOR dysregulation causes or contributes to a wide range of human diseases. And so in particular, if mutations cause TORC1 to become hyperactive, that can lead to uncontrolled growth, formation of tumors, cancers, and other kinds of diseases. And so there are indeed mutations in the TOR kinase, in the kinase domain that cause it to be biochemically a more active kinase, and those can be oncogenic themselves. And in nearly 100% of cancers, there's some mutations happening. If TOR was working properly, the cancer would not be able to develop in the first place. So there's a huge industry, hundreds of labs probably that are working on TOR signaling in mammals, which has resulted in a really detailed understanding of the signaling network that acts upstream of TOR. So this is a recent review that is considered to be a real oversimplification of what's happening upstream of TOR, of how TOR is monitoring some nutrients in human cells. And I say it's an oversimplification. You can see there's a lot of components and complicated arrows here, but this is really just the narrowed down view. And now I'm going to gray out everything that is not conserved to plants. And you can kind of see the color's not great there, but you can see that almost everything goes away. So this upstream network that's coordinating TOR signaling is not conserved between mammals and plants. There are two hypotheses that we can think of to explain this. One is that plants have just evolved a totally different set of signaling networks to control TOR signaling, to monitor nutrients. And of course, that is somewhat true. The other explanation is that what we put into the reviews and what we view as the key components upstream of TOR is not actually the ancestral conserved upstream functioning of TOR. And I'd be happy to talk about that more, but we also see quite a bit of that what's conserved is often multifunctional proteins that signal to TOR, but also do other things. There's not a lot of genetic variation in them, so they're not great druggable targets because they're doing too many things in the cell. There's not a lot of genetic variation, so they don't come up in like a GWAS study of human populations. They don't come up very often in screens of cancer. And so by instead of taking this comparative approach, we're starting to learn a lot more about some of the core components that are really crucial for signaling to TOR. So just to summarize that with my little cartoons I drew of different species, because I thought that was a fun thing to do one day. This was peak COVID. I decided I was going to draw all the models. There's so many more than these that I've shown here. Just at home with my husband drawing models. But so on the left-hand side, we have the animals and the fungi and even slime mold in which TOR signaling has been really well studied. In plants, we're just beginning to get there with more and more labs taking interest, especially in algae and in the model system Arabidopsis. Almost nothing known about early diverging protists, and I might get a chance to talk about that at the end. So in addition, most of our funding comes from the NIH, and so usually my selling point is about using plants as a sort of biomedical model for evolutionary comparisons. But we're also really interested in agriculture, and I'm just going to give my quick doom and gloom story. So just strap in for a second. Global warming, blah blah blah. Okay, so I think that many of you are familiar with the story of the Green Revolution, which was effectively add fertilizer to soils, and you get higher yields. And so in the 60s and 70s, this really revolutionized agriculture around the world. The US puts as much fertilizer as we possibly can, more than we need to, on all of our soils. And we have done so since the 70s. China has just hit the point where they're putting as much as they can on all their arable land as well. India is catching up, Africa is catching up. And that's really been necessary to sustain the agricultural system to supply food to billions of people. There are many different issues with this. I'm just going to highlight two. The first is that fertilizers are a finite resource. And I like to focus mostly on phosphate because this is the most finite of the resources. Phosphate is a rock that we mine from the earth. Once we apply it to soils, it gets into the water system and we have no way of recovering it. And phosphate mines are expected to start running low in the coming decades, maybe even starting to run out in the coming, optimistically century we would be, but could be less than that. We don't really know. And so there's no plan for what we're going to do when we start running low on phosphate. And then the other one that I don't have to tell people about in Wisconsin is runoff and the effects that runoff has on our waterways and leads to all kinds of ecological problems and is basically one of the major contributors to greenhouse gas emissions. And so estimates from people who are more economists and social scientists have said that some of the simple things that we think of, like dietary changes, waste reduction, and so on, if we all became vegetarian, I say that's simple. If everybody became vegetarian, that would not be enough to move us to a sustainable agricultural system. But maybe with technological and especially biotechnological improvement of plants that they more efficiently use fertilizers, we'll be in a situation where we can actually sustain agriculture. So my hope is that by studying this core conserved pathway that monitors nutrient availability and then controls growth, that we'll be able to contribute to engineering plants or breeding plants that have improved nitrogen use efficiency for a sustainable agricultural future. And I'll show a little hint of that towards the end of my talk. Okay, part two. So I'm going to tell you a news story that I have never talked about in public before, partly because Worf forbade me from talking about it in public before, but now I'm allowed to. So about an amino acyl tRNA synthetase that stimulates TOR activity in plants. Like all good stories, it started with a forward genetic screen. The forward genetic screen here in Annihilation is Natalie Portman and friends walking into a giant bubble where mutagenesis is happening like crazy. And inside that bubble, Tessa Thompson finds these plants that have evolved to start looking like people and other kinds of weird shapes. And some of the plants are eating people and terrible things. And it's a great movie. I have to briefly introduce plasmodesmata, or PD, which are nanoscopic channels in plant cell walls. Plasmodesmata are membrane-bound. They connect the cytoplasm of neighboring cells, and they allow for all kinds of molecules to move between plant cells. So small things like metabolites, hormones, ions, water are all freely trafficking via plasmodesmata between cells. Larger things like proteins and small RNAs are also moving up to maybe proteins up to 80 kilodaltons. If it's in the cytosol, it can move between cells, depending a little bit on developmental and physiological context. And then viruses and other pathogens hijack plasmodesmata to support their virulence. So these are really critical structures to plant biology. There's no such thing as a plant without plasmodesmata. It's evolved repeatedly, independently in any lineage that has complex multicellularity with a cellulosic cell wall. So this is just like a really critical aspect of plant biology. And investigating plasmodesmatal function has been really challenging because of how much they're doing. They're doing so much. It's like saying, I want to study what a plant is like without a nucleus. It's a core part of the cell. So my PhD advisor, Pat Zabriskie, set out, actually, this started a little bit in the 90s, to do a screen for mutants that have defective PD trafficking. And there's two aspects of this screen. One is the assay that I'll tell you about in a second. And the other is that Pat was coming in, not a plant geneticist at all, but had been hired by a plant biology department because of her work on agrobacterium. And she said that she only wanted to work on lethal mutants, that she didn't believe that it was worth studying anything that was alive. If it doesn't die, who cares? Not that big a deal. And in fact, my students can tell you that to this day, if you want to get my attention, you have to show me a dead plant, and then I'm interested. Anything short of that, I'm like, I don't know, maybe it's not that big a deal. And then the assay was for two directions: for increased PD trafficking of what's shown here in red is chlorophyll, and then in green is a dye that's 10 kilodaltons, and it traffics really rapidly through this embryo. We call those ISE mutants. And then on the other hand, a screen for decreased PD trafficking, where a very small dye, pyranine, loads rapidly throughout the wild type embryo that can only slightly enter into this mutant that has decreased PD trafficking. There's me and Pat 15 years ago. The first part of that screen, what I did during my PhD research, was showing two of these mutants, ISE1 and ISE2, that revealed that PD transport is tightly coordinated with chloroplast physiology. And here I'm showing in green some chloroplasts that are reaching out with these structures called stromules, which are tubular extensions from the chloroplast, and they're touching the cell wall that's highlighted in pink. And I have other images showing that those are in fact reaching out and interacting, associating with plasmodesmata directly. And if you want to hear more about that, you can read some papers or you can come and talk to me, and I'd be thrilled to talk about that. But right at the end of my dissertation, we finished mapping the last couple of the ISE mutants. And then that was the starting point for when I started my group at Berkeley, was really pursuing these and showing that the other half of the ISE mutants identified in that original screen, the last two, are both defective in assembly of the TOR complex. And I'm not going to get into the details here because that's what I talked about during my interview talk, but just to say that both with forward genetics, so both ISE3 and ISE4, and then also with reverse genetics, we were able to show that assembly of the TORC1 dimer, which starts off as a protomer of 3 subunits and then comes together as a dimer, and an HSP90 and ATP sensitive pathway, that assembly is really critical to restrict PD trafficking during embryogenesis and also later on in development. This was such a powerful result because no forward genetic approach had really identified TOR signaling before. This was the closest we have to a forward genetic screen that leads to multiple mutants that are defective in TOR signaling through this PD trafficking screen. And so as I was finishing up my PhD, I decided that we should go back and extend the screen a little bit It's a really arduous screen. Each time you screen these, it can take hours to screen each line to get all of these replicated and so on. But so instead of doing an EMS screen, which is what Pat had done, which then takes forever to get through all the different lines, I decided to use tDNA lines that are available and take a more reverse genetics approach. And from that, I found a fifth ISE mutant called ISE5 that disrupts PD trafficking and also development. The people who are into Arabidopsis embryos here can see these are really screwed up embryos. So it's not really making cotyledons. Here I'm showing chlorophyll. So this apical growth is not actually a cotyledon. It's some other kind of growth. We see what looks like sort of broad expression of knotted 1 here and some other factors. And again, I'd love to talk, but I promised myself I wouldn't talk too much about development today. So anyone wants to talk about development, we can talk about that too. And so we looked to see which tDNA line this was. And it turns out that ISE5 encodes a tRNA synthetase. And I went and talked to Pat about this. I'd already moved on from I was in my postdoc, but I went and told her, oh, look, ISE5 is a tRNA synthetase. And she said, listen, Jake, I have to retire. This is too much. I can't handle shifting and working on a tRNA synthetase now. Good luck. And that was that. I think she's never wanted to hear about it again. So, so in order to think about how a tRNA synthetase might be affecting PD trafficking and indeed TOR signaling, I have to take a quick detour and talk a little bit about amino acid sensors for TOR in mammals. And before I get into that, I have to say that the best way that we have of measuring TOR activity, so it's a protein kinase, is by looking at its phosphorylation and activation of a downstream substrate or direct substrate called the S6 kinase. S6 kinase phosphorylates ribosomal protein S6, and so that's where it gets its name from. And so the way that we probe this, we raised monoclonal antibodies that detect the total S6K levels. Then we use commercial antibodies that are phosphospecific against S6K. And so when TOR is active, it phosphorylates this threonine. It's really well conserved across eukaryotes, except for baker's yeast, of course, because everything's always different in baker's yeast. But other than that, it's really well conserved. And then we can measure TOR activity by looking and seeing, so in this case, this is a plot that Michael did in his nucleotide study, and that I talked about during my interview, where you can see that the S6K total protein stays at the same level, but there's this shift in the phosphorylation of S6K from when TOR activity is high, it's very phosphorylated, to when it's low, it's very not phosphorylated. That's just to orient us a little bit. And this has been the major assay for studying TOR signaling in mammals and plants for quite a while now. So in mammals, there was this finding after Joe Heitman discovered the target of rapamycin and people started working on this in mammals, they decided to test. They knew that amino acids were somehow involved in stimulating TOR. And a bunch of groups did a bunch of different experiments, and I'm just going to show you one result that's kind of clean. But everybody found basically the same thing. If you remove amino acids from the serum of human cell cultures, S6K is extremely inactive. If you add back in all 20 amino acids, S6K activity goes back up. And then they tested adding each different amino acid on its own. And consistently across different cell types, different experimental conditions, and so on. Leucine is the amino acid that strongly activates TOR and leads to activation of S6K. And that activation is entirely dependent on TOR. If you add rapamycin to the system, leucine is not able to activate this anymore. Now, just to say, and it'll come up again, humans are such, humans are these simple lower organisms that they can't make amino acids for themselves. Everything is so straightforward. Hiroshi's here, he can tell you plants, higher organisms like plants that can make all 20 amino acids for themselves. And it's much more complicated. So this experiment's pretty easy to do in human cells, a lot trickier to do in plant cells. And so, in fact, there have been similar attempts to do these kinds of experiments in plants. We tried them as well. And basically, depending on how you do it, you can show that any of the 20 amino acids can stimulate TOR in plants. And that's because all of the amino acids, when you add them in, can basically be interconverted around and shifted around. And so I don't think that that's going to be a productive approach for understanding amino acid sensors in plants. Then just to show you a little bit of how this work has built in the last 10 years or so, some of the direct amino acid sensors for TOR have been identified in humans. And I'm just going to show two stories that I think illustrate evolutionary paths to becoming an amino acid sensor. And the first one is a leucyl tRNA synthetase that, when it's bound to leucine, stimulates TOR activity. How it does that is hotly contested. This is the kind of thing people get really heated about, and so I'm not going to talk about it, but just to say leucine stimulates TOR through a leucyl tRNA synthetase. So you can see that's the connection that's going to come up in a moment with our story about an asparaginal tRNA synthetase. In another pathway, there's a protein called cestrin-2, which evolved quite a long time ago in the lineage of fungi and animals and others. And cestrin-2 is activated by the transcription factor p53 that many of you are familiar with. That's a stress-activated transcription factor. When p53 is active, it induces expression of cestrin-2. And cestrin-2 then inhibits TOR to arrest growth under stress conditions. But when leucine is very abundant, there's a binding pocket on cestrin-2 that leucine can bind to, and it actually causes this conformational change that I drew those off of the structure. So it goes like this, and then it can no longer inhibit TOR. And so we have a case where there's a protein that already knew about amino acids, leucyl tRNA synthetase, and then evolved a mechanism for activating TOR. And then we had a protein that was already involved in regulating TOR and later evolved the ability to monitor an amino acid. And so that's just to say there's kind of a couple different ways you conceptually think about this. And I call those adaptation and exaptation. Okay, but as I showed before in a different way, this is a drawing from one of my PhD students, Nanticha. On the left is the pathway in humans, and on the right, and so again, a little grayed out anyway, but on the right, what's illustrated is that basically the whole pathway that's upstream monitoring monitoring amino acids in mammalian cells is just not conserved to plants at all. Almost all the components are just missing. There is a leucyl tRNA synthetase, of course, but it's not doing anything to do with TOR as far as we can tell. So, as I said, plants can synthesize all 20 amino acids, and there's a lot of interconversions. It's actually really hard to draw the amino acid biosynthesis pathway because it's all circles and loops that are going back onto each other. And so we usually end up drawing it, showing it this way, but all of these lines end up circling back and around. And so trying to figure out which amino acid might be activating TOR using a more physiological approach, I think is just not ever going to really work out. A genetic approach, though, you know, not to oversell my point, but genetics seems to be a powerful approach here. Nonetheless, just to show that indeed nitrogen and amino acids can activate TOR in plants, we grew another model system, Nicotiana benthamiana, on substrate, and we added nitrate at various concentrations. And as you can see here, as we start to add nitrate and the plants start to grow, TOR activity skyrockets. And so indeed, TOR is monitoring nitrate, and we can do similar things with amino acids. So, okay, so that was my detour through human amino acid sensing, and now I can go back to ISE5. So, ISE5 encodes an asparaginal tRNA synthetase, which I'm calling NARS1. It's a class 2 tRNA synthetase, which forms this kind of structure where there's a dimer of tRNA synthetases that interact with tRNA, shown in orange and magenta here. And tRNA synthetases catalyze two reactions. First, they catalyze the amino isolation of the amino acid. So the amino acid interacts with ATP, and the ATP is hydrolyzed, resulting in amino acyl AMP plus pyrophosphate. And then the second reaction is condensation of the amino acyl AMP with the tRNA, releasing AMP. So those are the two reactions that this is catalyzing. In Arabidopsis, there are 4 genes that putatively encode asparaginal tRNA synthetases. NARS1 is the gene that ISE5 disrupts with a tDNA insertion. NARS3 is a very closely related paralog, has the same gene structure, 95% identical amino acids, basically the same thing, much more lowly expressed, maybe 10% the expression of NARS1. NARS2 is annotated everywhere as an asparaginal tRNA synthetase, but it's not. It can't be. It's missing all kinds of key residues for that. So structurally, it looks like that, but it's doing something else. And we're really interested in that, although the mutant doesn't have a very clear phenotype. It does have a little bit of a phenotype. So, and then there's a fourth one, which is the asparaginal tRNA synthetase that's targeted to both the mitochondria and the chloroplasts, which people in my lab have named McNARS, because that makes them laugh every time they say it. And the mitochondrial chloroplast McNARS is just kind of, yeah, it's a standard enzyme that's going there and supporting genome expression in the chloroplasts and mitochondria. For those of you who are more structurally inclined, I'm just showing here that asparaginal tRNA synthetases have this N-terminal tRNA binding fold. They have in orange here ATP, AMP binding tweezers. They're arginine tweezers that this is, I'm sorry to the biochemists in the room. They like squeeze the ATP and then they like kind of does this. That's my understanding of what's happening. So yeah. And then in blue are shown the key residues for asparagine binding. So it's this pocket where asparagine is binding and then interacting with ATP. Here I'm showing the human and the plant NARS structures, which you can see are at least superficially very similar to each other. And indeed, they do align very well, except for one thing, which is that plant NARS one has this extra whole chunk that's shown here in magenta, which we didn't even realize for long. We sort of thought, oh, we were sort of ignoring it. And then we decided we should focus in and figure out what that chunk that wasn't aligning was and see if it had a name. And it does. It's called a WEP domain. WEP domains are named after the tryptophanyl, histidyl, glutamyl, propyl tRNA synthetases in humans that all have this domain. WEP domains are basically two alpha helices. This is a prediction from AlphaFold of the structure that are found in a lot of tRNA synthetases in different parts of the tRNA synthetase. They come and they go. Sometimes they're there and then you look in a species next door and it's not there anymore. We have no idea where they're coming from or where they're going. It just popped up early in land plant evolution in asparaginal tRNA synthetase out of thin air. If there's an evolutionary person here who wants to work on that, please come join the lab. I would be thrilled to have someone figure out where these things are coming from and going. It's crazy to me that the same structure appears repeatedly over and over again and then just disappears and appears and disappears. What I can say is that we have no reason to think it's involved in any enzymatic activity of the protein because the protein has everything it needs for enzymatic activity already. We're validating that with in vitro, enzymology type assays at the moment. So now I'm going to show you about the connection to TOR. A lot of this is work from Nanticha Lutt, who did hundreds of Western blots post-pandemic. Since I came here, she did hundreds of Western blots mostly with her model, her favorite model, Nikoshiana. And so I thought she deserved a big picture because, and she would bring the Western blots to me and I'd be like, let's do that maybe 5 to 10 more times, and then I'll be happy with you. So I kind of tortured her, but she ended up with these beautiful results. So here is a situation where she looked in leaves where she had a mock agroinfiltration, or she agroinfiltrated to overexpress NARS1 or to overexpress NARS2. And you can see that NARS2 is not doing anything to activate TOR. And in the mock condition, there's also no activation of TOR. Actually, TOR is active. It's just we can't get these things often in the same blot without blowing out one of the bands because it's so different. So there's this incredible activation of TOR when we overexpress NARS1. And then just to cut to the chase, when we get rid of the WEP domain, which is that extra piece that's hanging out, that completely prevents NARS1 from activating TORC1. And so here I'm showing one of the dozens of plots of this, where you can see with NARS1, S6K is highly phosphorylated, and with a variant that's removing the WEP domain and actually then perfectly aligns with every other asparaginal tRNA synthetase across all of life, and so it should be perfectly functional, not able to activate TOR anymore, and so on. So these results are really clean, and so now we're really working mostly hard to make sure that we really do know that this WEP domain isn't doing anything catalytic, but it suggests that the WEP domain is involved in signaling instead. In humans, it's known that the tryptophanyl tRNA synthetase with a WEP domain, that WEP domain interacts with a kinase that's in the very small family of kinases that TOR is also a member of. And so we have some model to work off of there that this WEP domain could even be involved in direct interactions with the TOR kinase, because that's something that WEP domains apparently can do. Nanticha also made mutant variants of NARS1 that are defective in other aspects. So a variant that's defective in dimerization partially removes its ability to activate TOR, but not entirely. Which is shown here. A mutant that's defective in the ability to bind to asparagine. sometimes completely prevents. So here's the mutant that can't bind to asparagine very well. Sometimes completely prevents TOR activation. Here's another residue that's supposed to be important for asparagine binding. Basically no effect. These spots are all looking pretty similar. So we're also doing a lot of work to try to understand these predictions that we have bioinformatically. Are they actually impacting asparagine binding? How might they be impacting the signaling to TOR? And then this is the coolest result, I think. So Nanticha made a slew of transgenic lines overexpressing NARS1. And for those of you who are at Joe's talk, you want to talk about epimutations. The number of epimutations we have when we're making transgenic plants, it's all the time. So that was just kind of, it was like, oh yeah, of course there's epimutations. Duh, that's what happens. So that happens out here. But when we actually have lines that are good at overexpressing NARS1, you can see here, these are seeds that germinated at the same time, same age seeds, everything else was the same, and consistently across many different experiments. And I made Nanticha repeat this so many times because I didn't believe her. You can see that the seed, I mean, I believed her, but I didn't want to believe it. Didn't trust myself. You can see that the NARS1 overexpressing plants are growing faster and bigger as seedlings and as mature plants. And so you can see that this line is just going gangbusters. And it's not just one line, it's many different lines that we have that are doing this. And so speculatively, we think that this is due to hyperactivation of TOR. We've made lines with other tRNA synthetases that don't have any effect like this. And so we really think that this is something specific to the NARS1 gene. So why monitor asparagine? Does anybody know the origin of the word asparagine, why it's called asparagine? Asparagus, yeah, that's right. So asparagine was actually the first amino acid characterized biochemically because it's so abundant in asparagus that they could just grind up asparagus and get huge pure preps of asparagine out of it. And that's common not only in, I mean, asparagus is kind of an outlier, pretty extreme, but in general, many plants use asparagine to store and redistribute nitrogen. Asparagine is really stable, really soluble, has the highest nitrogen to carbon ratio of any amino acid. It's often the endpoint of fixation of nitrogen in the root and then is trafficked as asparagine, sometimes also glutamine to the shoot for further use. And then asparagine is rapidly interconverted into other amino acids. And just to say, we thought we should at least show this. Indeed, if we add asparagine to plants, it does activate TOR. So if we grow Arabidopsis seedlings in the absence of nitrogen, and then we add asparagine, it rapidly activates TOR, as measured by S6K phosphorylation, in a situation where if we add glucose, it doesn't do anything, basically. And then we also have a dose response curve here showing that as we add more asparagine up to a certain level, if we add more than 20 millimolar, there starts to be toxic effects. But around there, we start to see this nice curve showing that asparagine is activating TOR. So our working hypothesis is that NARS1 is the first putative cytosolic amino acid sensor in plants and that it's monitoring amino acid levels for TOR. And a lot of the work going on in the lab now is to try to understand the signal transduction pathway from NARS1 to TOR. We have a little bit of evidence that it may actually be directly interacting with the TOR complex and lots of evidence that there's at least an intermediate that TOR is touching and that NARS1 is touching. So a lot of exciting work to come from that. Okay, so there's Natalie Portman looking, she's so curious about this flower, which I can relate to that. You see a cool flower and you're like, oh my gosh, how does that, and that's, I mean, this is my career. It's just me saying, what's going on with this weird flower? So just to throw that in there. All right, there's no reason for that slide. Lastly, I only have a couple minutes left and I want to make sure that I leave some time for questions. So I'm just going to zoom through for a second and tell you a quick story about the other side of TOR signaling. I've been talking about what's happening upstream, but a lot of the lab is working downstream of TOR using multi-omics approaches to map TOR-sensitive signaling networks in plants. And we're especially interested in how TOR is promoting ribosome biogenesis. We see really clear evidence that ribosome biogenesis and ribosome abundance is tightly controlled by TOR signaling and nutrient availability, and we want to understand better how that's happening at a mechanistic level. And so to do this, we've taken advantage of an assay that was developed by Jen Sheen's group at Harvard, where she germinates seedlings until they become quiescent and they've used up all of their maternal nutrient stores. And then under the right light conditions, and they're on a little bit of liquid, so they're not hypoxic, but they're a little hypoxic and so on. They become quiescent, TOR is inactive, and if you add glucose to those seedlings, it stimulates rapid growth that is TOR dependent, and like 99.9% of the transcriptional changes induced by glucose require TOR. And so this is a really acute assay for specifically activating TOR through glucose. And then we can look at different time points and under slightly different conditions to try to understand how TOR engages downstream effectors. Yeah, I will say in the phosphoproteome that we've done a lot of phosphoproteomes of this, and there it's 99% of the changes are TOR dependent. And then there's this one protein that is very clearly TOR independent, and we think is acting in a signal transduction pathway from glucose to TOR. And that's going to be the topic of Katie Lund's PhD project. So more to come on that eventually. So we do some various different assays where we add glucose or TOR inhibitors. We can use chemotherapeutic TOR inhibitors that have been developed for humans. They work just as well in plants as they do in mammals, and under various different conditions. And then we do as much profiling as we can. So we look at transcriptomes, translatomes using ribosome footprinting and polysomal RNA sequencing, proteomes, phosphoproteomes, metabolomes. You know, if anybody has another ohm that you'd like me to add in there, I'm delighted to add more ohms. It just kind of gives me more data to look at. And I'm just going to zoom through and say that the major takeaway here is that at every level of genome expression, we see that TOR is promoting ribosomal protein biogenesis. So TOR promotes transcription of ribosomal protein genes. TOR is regulating splicing of ribosomal protein genes. TOR is promoting the translational efficiency of those ribosomal protein mRNAs. TOR is promoting ribosomal protein abundance and stability. And TOR is phosphorylating proteins that contribute to ribosome biogenesis and or are themselves ribosomal proteins. And some of this has been published in eLife shortly after I got here. I also did make a mistake once, and I said, can we change the name of a couple of these ribosomal protein genes? Because the nomenclature was a little messy in plants. And then I ended up leading this international committee to rename all. So I've renamed all of the ribosomal protein genes in plants. So if anybody is publishing in plants on a ribosomal protein gene, please check the nomenclature so that, because I get emails from SwissProt and Uniprot at least weekly without saying, could you please tell these people that they need to update to your nomenclature? And by the way, do you think this gene in whatever species that just got sequenced, do you think it's this one or that one? And it's a disaster. And I'm going to skip over a story about an RNA binding protein called LARP1, which does not stand for live action role playing, but could, but it said is an RNA binding protein that binds to transcripts that start with a stretch of pyrimidines called 5' top motifs. And basically, this is a case where it turns out that a pathway that's poorly understood in mammals turns out to be the ancient conserved pathway. So in mammalian systems, it was thought that translational regulation of these transcripts was through various different mechanisms, and we've shown that this is actually conserved through the TOR-LARP1 signaling axis. That's published, so you can read about it. And now we're moving beyond Arabidopsis, looking at some other systems. So Michael has been working since he got here really hard on doing some massive multi-omics experiments in maize. He developed a similar assay where we can acutely, completely fine-tune TOR signaling in maize. And he has 16 different experimental conditions for which he's done RNA-seq and riboseq and proteomes and phosphoproteomes and metabolomes. And we have been learning so much from those data that we're now going to be pursuing for forever. And he'll probably be on the job market in a couple of years, and he can also go and pursue it. And we'll just have infinitely many questions to ask about how TOR is controlling development. And then lastly, just to say that we're also branching out a little bit beyond Arabidopsis and maize, because as especially the students in the room know, when I just love letting students do things that they get excited about, which does lead to sometimes broadening of the lab scope. But Ryan Martinez, shown there in his igloo that he made on the lake, he is also Californian, so he also was kind of in shock about the whole winter thing. But he's been working on the functional genomics of a plant from Australia that makes very few ribosomes, almost no ribosomes compared to any other plant that we know. And so he just sequenced and assembled genomes, transcriptomes. We're going to be looking and seeing how these plants express their genome with as few as 40-fold fewer ribosomes compared to any other plant that's been studied. And then Katie Klimple decided to start on a new project studying this early diverging protist called Naegleria that diverged from eukaryotes before the split between plants and animals. So really like a dark ages period of eukaryotic evolution. And it has TOR and a couple of accessory proteins. And other than that, kind of a big empty space of what we know. And so she's working on that. So there will be some exciting stories to come in some other species as well. And thanks to collaborators and so on and so forth, and funding, mostly from NIH, but from some other people too. And thank you guys.
I was just wondering if
That's okay, yeah.
I was if you always NARS1 in place, did you titrate the amount of nitrate in the soil to see if they can grow with lower levels of nitrate more? And is it lethal if you have no nitrate at all in the soil? Because and what is that?
Yeah. So Danielle's asking, when we are overexpressing NARS1 and it's causing this uncontrolled growth, how does that relate to the amount of nitrogen it's taking up? And the short answer is we don't really have strong experimental evidence one way or the other yet about that, but it is something that we're pursuing. There are a lot of complicated components in there, including, I mentioned the thing about epimutations. These lines tend to not be very stable in that they are triggering post-transcriptional gene silencing fairly often. And so we end up with sets of classes of lines where some are really big and overexpressing NARS1, and then some have NARS1 completely knocked down and the plant can barely grow. And so we're working on getting lines that we think are stable enough that we can really do those experiments, hopefully with a range of NARS1 expression. Right now we kind of have a couple lines that are really strongly expressing. We'd like to have a few more before we get too lost in that. Yeah.
You probably already thought about it, but if I wanted to apply this transgenic plant overexpressing NARS, right?
Yep.
They would behave differently in different types of soil.
Yeah.
Have you tested? But are you interested?
Yes.
Okay.
Yes, we will have done that. And I should say, so I showed the Arabidopsis data. We also have overexpressors in Nicotiana benthamiana, in soy, and in maize. And we have some similar results in all of those. And so I kind of want to have all of my ducks in a row before we start doing some of those more practical experiments, because there's going to be a lot of troubleshooting to get those experiments working cleanly. Rick.
It's just related as well. Your answer to the last question.
Yeah.
Does that mean you're interested in looking at the nutrient environments for optimal growth of wild type versus NARS1 overexpressed?
Yes.
Okay.
Yes, absolutely. I mean, I think the question is partly if these plants are growing so much bigger and faster, they must be using more nutrients to do that. But is there some trade-off that we can hit there where we're using less nutrients and getting more growth? That would be the fantasy. Yeah. Yep. Chris.
Yeah, so you made a couple of interesting comments about plasmodesmata. First, that they're fundamental to plant biology.
Yeah.
But also that they evolved independently several times in plants. What's known about the molecular mechanisms of how that evolves, the similarities and differences are in different languages?
Yeah, so in land plants, it's a synapomorphy. All land plants have plasmodesmata that evolved once and have a specific structure that includes endoplasmic reticulum inside the plasmodesmatal structure. In multiple other parenchymatous green algae that have evolved multicellularity, not just single files and not flat thallus, but if they start to have three-dimensional multicellularity, repeatedly in many different lineages, structures that are analogous to plasmodesmata have evolved that look extremely similar to plasmodesmata, but don't have that same strand of ER in them for the most part. And so, and some of them have other structures that we can see ultrastructurally. Those algae are all strange marine algae that nobody really studies in much detail, so we don't have much functional information about those. But it's thought that the sister to land plants at the moment is a unicellular or maybe a phyal, single stretch of algae. So all of those other algae that have something that looks like Plasmodesmata, it evolved in parallel, or you could get into a debate about that, but yeah. David.
How do you think the amino-acyl tRNA synthetase, when it's bound to the amino acid, activates TOR?
Right. I'll tell you my fantasy. And it is based on this information that we have from a tryptophanyl tRNA synthetase in humans from work from Paul Schimmel's lab at the Salk Institute. So what he showed, and what really, I've done a little bit of modeling stuff, but I'm not an expert at it, and so I don't. If I'm answering a question, I'm not saying what I think is this. I'm fantasizing. It seems that this WEP domain it's on extremely flexible linkers, and so depending on how you model this could float around different places. But I like to think that when asparagine is present, the WEP domain bends over and is interacting in some way with that asparagine. That actually, you can make a model where that's happening and it's interacting with the asparaginal AMP. And that is the case with tryptophanyl tRNA synthetase, that's what happens. The WEP domain goes, whoop. And then that shifts the protein-protein interaction that's happening. And so whether it's that the tRNA synthetase was interacting with something and then stops interacting, and that allows for activation of TOR, or if when it goes into this, it gives it a surface that allows it to interact with TOR, something along those lines is kind of what I'm
Do you think it's
speculating.
directly interacts with TOR?
Well, so these WEP, there's at least one, and there's a couple other hints of WEP domains directly interacting with the kinase domain of phosphatidyl and inositol 3 kinase-like kinases. So I think it's plausible. That seems too simple to be true, but it could be. I kind of, if anything, I kind of like to have a little something indirect so that I have more things to work on than just like, and interacts with TOR kinase and something happens. That feels abstract. But we are one of my graduate students is interested in doing some structural work at some point to try to really nail this down and see if we can see a change in the WEP domain. And then we're doing lots of proximity labeling and co-IPs and so on to see how this WEP domain affects protein-protein interactions with NARS1. Yeah. John.
Wanted to follow up on WEP domains.
Yeah.
It sounded like you're suggesting that they appear on different tRNA synthetases in different lineages.
Mm-hmm.
Do you think that might occur via some sort of non-homologous gene conversion between different tRNAs?
Yeah, I think that's a plausible explanation. The trick is that tRNA synthetases don't add things otherwise. Like there aren't other things that get added to tRNA synthetases very often. There aren't a lot of other domains popping up all the time. It's just this one kind of domain. So yeah. And then the other part that's interesting is that the WEP domain doesn't pop up in the same place all the time. So for instance, the glutamyl propyl tRNA synthetases, one tRNA synthetase ends, and then there's 6 WEP domains, and then the other one begins. And it's a conjoined protein. So I think that the explanation you propose, which is a convergence and not really a homology, is probably true, but that there's enough mystery here that I usually can sort things pretty easily and at least convince myself that I know how it evolved. And in this case, I think it's an open question.
And in terms of the consequences, there is the prediction that basically, makes it so different amino acids are involved more in TOR signaling in some species versus others.
Yeah, so and I guess that goes to something I sort of said obliquely, which is that I think that one of the lessons from the mammalian system is that there are a lot of different proteins that are sensing amino acids for TOR, and different amino acids, not just leucine, others as well. Leucine is the most potent activator, and there are probably a couple of major players for that in those experiments. But in an actual cell, there's lots of other proteins that are monitoring. And those weren't found through biochemical approaches necessarily, or through physiological approaches, but instead through protein-protein interaction studies for the most part. Here, I think that genetics is really helpful because we can find mutants that are defective in TOR signaling and then use that as a way to find these putative sensors. And so in this case, asparagine wouldn't have been maybe my top pick if I were just looking at the 20 amino acids and saying, who do I think plant TOR senses? Although it would have been close to the top because of its role in storage and trafficking and its dynamic regulation in response to conditions. But I think that the genetics led me here. But I don't think this is going to be the only amino acid sensor for TOR. I think it's just the first one we found. Yeah. Audrey.
It just occurred to me, and maybe this is not an I don't know. You mentioned that TOR can regulate the translational machinery.
Yeah.
And respond to nitrogen availability. Does TOR respond to translational to the cell? And what's the mechanism of that?
Yes, and many mechanisms are at play. The probably best studied is the stringent response, which across organisms is mediated by GCN2, which is a protein kinase that binds to GCN1 and GCN20. GCN1 and GCN20 bind to stalled ribosomes. And there's a little bit of debate about the structure there, but so when ribosomes are stalled, they activate this protein kinase GCN2, which then phosphorylates EIF2 alpha, I think, and that leads to rapid translational repression. But GCN2 is also phosphorylating other things that eventually have feedback onto TOR. TOR is monitoring in many other ways as well, translational efficiency. But I will say a fundamental experiment here in the mammalian field that we've sort of replicated in plants, but we need to do better at is if you add cyclohexamide, which causes ribosomes to just arrest in place, it leads to a transient hyperinduction of TOR, which is thought to be due to hyperaccumulation of amino acids in response to stopping translation. And so, yes, TOR is monitoring translation in a real cell over time, but that's maybe not the acute signaling pathway that's going towards a more complicated network. Does that kind of answer your question?
Yeah.
Okay.
I would imagine tRNA synthetases, they're required to charge tRNAs for translation, but I'm wondering, so that answered my question. I'm wondering if exactly the same mechanism could sense translation. If the tRNA
Yes.
the mechanism you just described makes sense, almost like a feedback on translation.
Yes.
Right?
Yes.
It could be lack of charged tRNAs, but could be something.
Absolutely. And I will say that we sort of assumed for a long time that this was going to be something enzymatic happening and maybe to do with a tRNA sensing mechanism. And the web domain data lead us away from that, which is much more convenient for us as well. And so I'm always cautious about that. So that's why we really have to nail down that the web domain isn't involved in enzymatic activity to show that this isn't disrupting tRNA synthesis, but is instead disrupting a different signaling pathway. Yeah. Anything else?
Any more questions? No?
Thank you.